Let's bring in glycine. And this is where glyphosate becomes a real problem, where the molecule, actually amino acid glycine, looks a lot like glyphosate, so it can sneak in. Talk about the mechanism there.
That is really crucial, I think. I think it's the really critical reason why glyphosate is so amazing in its properties, is because it's a glycine analog, which means it's actually a complete glycine molecule. The only difference is that there's a proton that's been taken off of the nitrogen atom, and then a whole extra piece that has been stuck onto the nitrogen atom to replace that proton. And that piece is a methylphosphonate. So it's a methyl group, which is CH3, but it has a phosphonate attached to it. And phosphonate is like phosphate, but one short on the oxygen. So phosphate is like PO4 and phosphonate is PO3. So it's got a whole bunch of extra stuff there, the methyl group and the phosphonate. The phosphonate is negatively charged. It greatly changes the properties of the molecule by virtue of that extra material stuck on the nitrogen atom. But it's still glycine. The whole thing is glycine. And so it actually matches, it goes where glycine goes, and it disrupts glycine everywhere it goes because it pretends to be glycine. But then it has that extra material which messes up everything. So what I theorize in my book, and it's actually a central story in my book, is that glyphosate is getting into proteins by mistake in place of glycine during the coding process. So you see the code for glycine, the assembly machine, and the machine is very elegant. It has a specific code for each one of the amino acids. So the code for glycine has to fit glycine inside a little area that's very small. Glycine is the smallest amino acid. So that particular protein that is the one that matches glycine, that grabs glycine to put it into the code, into the assembled protein, glycine fits into this little socket that it creates. And so does glyphosate, because the nitrogen has to stay outside of the socket because it has to hook up to the chain. So the part of glyphosate that is glycine fits into the socket beautifully. None of the other amino acids do. So it's how it can select for glycine, but it can also select for glyphosate. So glyphosate gets in there as long as there's not really bulky amino acids next door, so that if there's room for that methylphosphonate. And the situation where there typically is room is in the place in the enzyme where it binds phosphate. So phosphonate and phosphate are very similar. Glyphosate can put its extra piece into the slot, into the hole that's reserved for the substrate, because the substrate has a phosphate that needs to fit in there. Glyphosate can take advantage of that space to put its own phosphonate there, and that completely blocks the ability of the substrate to bind, which completely ruins the protein. And there are lots and lots of proteins that bind phosphate, as I showed in my book. I keep discovering more, and it's amazing. They typically bind phosphate at a site where at least one glycine residue is highly conserved, and that means that glycine is critical for the protein to work. Even when you just change it to alanine, which is a very small change, just an extra methyl group, it can be completely destroyed in its ability to do its job. And so glyphosate, it goes in there instead of glycine and turns into a bulky, completely different molecule, negatively charged, and completely disrupts the enzyme's ability to do its job. So you can just replace one glycine in one of these molecules, one of these proteins, with a glyphosate, and you can ruin its ability to do its job. And so I think there are glyphosate bullets all over the body that are glyphosate molecules stuck inside proteins where they shouldn't be, and messing up those proteins' performance and causing disease.
Okay, this is really important. I want to go even deeper here. To make sure I understand, we take in glyphosate in the body. When we're making new proteins, the cell makes a mistake where there would be a glycine and puts the glyphosate in there, which is going to change the protein, change its function. So are the proteins detectable by us? Can we look at a human body right now and see how many of our proteins have this change?
There is actually a whole technology that allows you to do exactly that. And it turns out there's a group of researchers who published a paper where they did that. And they actually looked for glyphosate substitution. And it's pretty cool technology because you can actually weigh the protein. And if it's going to be heavier if there's those extra pieces from the glyphosate in there. And they know exactly how much heavier it's going to be. So they can determine whether there is a glyphosate substitution for glycine in the protein that's produced by virtue of that extra weight. And so there was a paper published on that, and that paper said something like, the title was something like, "Glyphosate does not substitute for glycine during protein synthesis," something like that. The title does not. But when you read the paper, you say, oh my God, these guys actually found it. And they have a figure 2, I think it is. They have this big picture of these little boxes, and each box has a particular protein that where they detected glyphosate substitution at certain glycines. And that in each box has in tiny, tiny print above it, the sequence that in that amino acid sequence that they found where that glyphosate was there because it was too heavy. The glycine was too heavy by exactly the right amount to be glyphosate. And they had 12 of these, 12 different amino acid sequences, which I basically wrote down and looked up because you can look them up and find out what protein they're in. And I wrote a little, I wrote an article and put it up on my website where I critiqued that paper and showed that those particular proteins, 9 of them were binding phosphate. I mean, they were really beautiful examples of my glyphosate substitution theory, that the places where the glycines were being substituted made sense to me for those proteins. And then what I felt was that the experiment was, so they claimed that everything was a mistake. All of it was noise because they found kind of equal amounts in both the treated group and the untreated group. So and what they were treating was cancer cells. And these cancer cells had been maintained in culture for decades. They came from an ancient person a woman who had breast cancer. And they've maintained, these cells are famous. They're used in lots of studies. So they've been grown in culture for many, many decades. And when you grow them in culture, you feed them nutrients that are almost certainly contaminated with glyphosate. So these cells have been, both the treated and the untreated group have been exposed to lots of glyphosate throughout their many decades of living in a culture. And they've both picked up lots of glyphosate molecules and stuck them into various proteins. And then when they're grown in the experiment, they break down those proteins, release the glyphosate, and put it into a different protein. So they make new proteins and put the glyphosate in there that they got from the previous protein that was broken down in the whole process of living. So I think it's expected that the untreated group would have also lots of glyphosate substitutions. And so my feeling is that they didn't trust their data because both groups had equal amounts, but they should have trusted their data because the data is really striking in terms of which particular sequences where glyphosate was found as a substitution makes a whole lot of sense.
Given what we know now about what you just said, do we have any idea, percentage-wise, proteins in our body that might be affected by this?
Yeah, percentage-wise, it's hard to say, but I do think collagen is one to worry about because collagen has huge swaths of glycine. It goes for a long sequence of GXY, 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 where every third amino acid is a glycine. Tremendous opportunity for glyphosate to substitute. And there are various diseases, genetic diseases, that are caused by a single glycine in collagen can be substituted by something else to cause these genetic diseases, that the disease that has, like, where you can fold your fingers way back, really flexible joints, a condition, I forget what that's called, but
I can't pronounce it. Ehlers-Danlos syndrome? Something like
right.
that?
Ehlers, I know, EHLER, Ehlers-Danlos syndrome. Thank you. I'd forgotten that. I wrote about it in my book. So, so that one is actually going up in prevalence. Even though it's a genetic disease, so that doesn't really make sense. But if it is, in fact, glyphosate substituting for glycines is going to cause a similar effect. And so, especially if you already have the genetic predisposition, you're going to be more likely to have trouble early if you're also being exposed to glyphosate. Ehlers-Danlos, that's right. Yeah.
The reason I'm so curious, it sounds like a lot of our proteins in the modern world would be affected by this, given the environment. How functional are those proteins? If we're now accumulating all these hits over the years and it's so pervasive, how well are these proteins working in our bodies?
Well, this is where it gets really, really complicated. And this is why I think glyphosate is causal in so many. People argue back with me. I have all these correlations that are going up, all these diseases going up dramatically, exactly in step with glyphosate usage. And then people fire back, a correlation doesn't mean causation. And then they say, how could one chemical cause so many diseases? Right? Those are the two sort of pushbacks on that claim. And I think both are answerable because glyphosate's a mechanism of toxicity is so broad. and so widespread in terms of the damage that it does throughout your body, that I can believe it could cause all those different diseases by virtue of affecting all these different proteins that it affects. There's a huge list. And I can give you one example, which is the protein. There's a contractile protein called myosin. And myosin in the gut, myosin has, I think, 11 glycines that are highly conserved. Whenever it's highly conserved, that means it's important. So I do that. When I do my search, I look for highly conserved glycines, and then I look for their environment to see if they match my, whether they bind phosphate. I have a whole list of things I look for to find a target protein that I suspect would be especially susceptible to glyphosate, and myosin definitely stands out. And so there was a study on myosin, and there was a single glycine that they looked at in myosin, one of the highly conserved glycines. And they experimented with substituting alanine for the glycine. Now, that's the smallest change you can do. It has an extra methyl group. And then they discovered that when you have a version of myosin that has alanine instead of glycine, it can only contract at 2% capacity. 2%. So that is just really surprising, amazing, really, that small change can have such a big effect on it. And so they also showed that if only 2% of the versions of that protein in a muscle fiber had that problem, then the whole muscle fiber can only contract at 50% capacity, because they sort of get in the way of the guys that are still working. So glyphosate, doesn't wreck every single molecule in there. It only wrecks some. But if it were to disturb 2%, it would cause the myosin, the muscle fiber, to only be able to contract at 50% capacity. So that's pretty huge, and then if you look at gut paralysis, there was a study on a woman who tried to kill herself by drinking a glyphosate formulation. And then they had an article to look at what were the effects of that on her body, to try to understand what is it doing. And one of the things they pointed out was very striking, that it caused gut paralysis for this woman. And that's probably because it messed up the myosin so drastically that her gut became paralyzed.
For somebody, which is all of us by what we're saying here, who's incorporated glyphosate into our proteins in our bodies, what's the best we can do at that point? Once it's incorporated,
it's really
can we get it out of the protein? I assume not, but what do we do?
That's what's really hard. I mean, I think it completely, it continually recycles in your body. Like you've got a protein that's got some glyphosate in it, and then that protein, proteins get recycled all the time. So that protein goes into the lysosome, gets broken down into individual amino acids, and then there's your glyphosate free again. And now we can go find another protein. So I think the glyphosate is constantly shuffling around into different proteins in your body, causing different kinds of disturbances all the time. So you've got glyphosate bullets all over your body if you've been living with glyphosate for decades, which we have at this point. And I think we all have lots of glyphosate scattered throughout our bodies in various proteins, ready to do some more damage with some other, if you get unlucky and it happens to go into a particular protein that has devastating effects. Even, for example, Alzheimer's disease is pretty amazing, because there's this GXXG motif that's a characteristic feature of proteins that are associated with amyloidosis. So there's there's papers on Alzheimer's focusing on this particular sequence in amyloid beta, which is the protein that's misfolds in association with Alzheimer's, that has GXXXG sequence, and those glycines have been singled out as being critically involved in the misfolding process. So if you replace those glycines the glycines are critical to form an alpha helix, which is the form it wants to have to be healthy. When you kill those glycines by replacing them with glyphosate, the alpha helix falls apart. It turns into a beta sheet, and that's the misfolded form of amyloid beta. So, and of course, Alzheimer's is going up dramatically, exactly in step with glyphosate usage. I think it's quite possible that misfolded amyloid beta, due to glyphosate substitution, is a major factor in that whole process.
It's obvious that we want to minimize or avoid glyphosate best we can. But since we're focusing so much on glycine, my brain goes to, can we increase glycine through supplementation or the diet to try and provide that substrate when proteins are made?
I think that's true. And in fact, I know a lot of naturopaths who have told me that they find glycine has been beneficial as a supplement for their patients, and they recommend it. And it makes a lot of sense, because if you sort of want to overwhelm the glyphosate with so much glycine that the machinery is less likely to pick up a glyphosate by mistake if there's plenty of glycine lying around. And I think that's well motivated. I think that's probably a very good idea. I definitely support a bone broth as a food, a really healthy, nutritious food. Bone broth made from, of course, grass-fed beef. You don't want there's a lot of glyphosate in the cows because they get a huge amount of glyphosate in their feed, so that's also a problem. Certainly eating certified organic to me is the #1 most important thing you can do to reduce your glyphosate exposure, not completely eliminate it, because it's still found in many samples of organic food, even though they're not allowed to use it.
If you enjoyed that clip, you're going to want to head over here and catch the full episode. I'll see you over there.
A lot of people don't know what glyphosate is, and I admit I didn't know what it was either back in 2010, 2011. They patented it to be used to strip metals off of pipes because it's very good at chelating metals. I think it was an accidental discovery.